Hi everyone, my name is Laura and I'm from the University of Toronto. Today I'll be presenting on one of my main projects for my PhD, which is called The Gradient of Developmental and Injury Response Transcriptional States Defines Roots of Glioblastoma Heterogeneity. In this project, we essentially use single-cell RNA sequencing to take a deep dive into cancer stem cell biology and glioblastoma. So glioblastoma is the most common and aggressive form of brain cancer in adults. Currently, therapeutic options minimally extend patient survival, and there's very high rates of recurrence. And this is partially due to the fact that glioblastoma is an extremely heterogeneous disease at the cellular, epigenetic, and genetic level. So this project focuses on tackling this issue of heterogeneity from the lens of a cancer stem cell. So glioblastoma has been shown to contain rare populations of cells called glioblastoma stem cells, which have the ability to self-renew, so recreate themselves, also the ability to differentiate to propagate the bulk of the tumor mass, as shown in orange here. And probably most importantly, they've also been shown to be extremely resistant to chemotherapy and radiation. So for this project, we are really interested in taking a better look at these cancer stem cells and glioblastoma to help us understand the transcriptional states and biology that might underpin their resistance to chemotherapy and radiation. So glioblastoma stem cells can be cultured directly from primary tumors. On the top here, I have a schematic of this culturing process that we do in collaboration with Peter Dirk's lab at this Sick Kids Hospital here in Toronto, as well as Sam Weiss at the University of Calgary. So first we get a fresh biopsy from a patient tumor, which is then dissociated into a single cell suspension. We then do our best to isolate the tumor bulk using density separation protocols, and then we essentially plate these enrichment of tumor cells in one of two ways. The first way is we plate it down into a dish, which is coated in laminin, which causes these cells to basically lay flat and form an adherent culture, which I'm sure anyone who's done some wet lab is really familiar with. The opposite, or the other way, is to basically plate these cells in the absence of laminin, which kind of causes them to grow as little balls, which we call neurospheres. So by using a specific neural stem cell media, what this protocol is able to do is basically get rid of all the tumor cells that are just the bulk progeny and really enrich for these cancer stem cells. So you end up with a more purified population of cells that express neural stem cell markers such as nestin, SOX2, and vimentin. And then also, if you inject them into a mouse brain, they're tumorigenic and can form a mouse. So using single-cell RNA sequencing, we wanted to ask if these cultures that we grow out contain a homogeneous population of cells, or if we find multiple different subclones or transcriptional states present in the GSCs that we derive from individual patients. So to answer this question, we profiled just over 69,000 cells, which were derived from 29 patient-derived GSC cultures. And as most single-cell RNA sequencing projects perform, our first step in the analysis was to basically place all of these cells onto one global map. So in this first plot on the left here is a t-SNE plot where each dot represents a different cell, and then the cells are colored by their patient ID or culture ID. So similar to almost all the other cancer single-cell papers, we can see that all these different cells cluster out separately by their patient ID, which highlights the fact that there's probably a lot of tumor-specific expression profiles that are being expressed in these GSCs, which is likely due to very different genetic profiles. And on the right here, we have the exact same plot, but instead of it's a t-SNE, it's a UMAP. And we can see that in some instances, when we were actually able to grow out different GSC cultures from different regions of the same patient tumor, that these different cells actually co-localize on the plot, showing that these GSCs from different regions of the same tumor are different, but they're more similar to each other compared to the cells that were derived from completely different patients. So next, we wanted to ask that when you look directly inside one culture from one patient, do you see different transcriptional groups? So to do this, we clustered each of our samples individually. So here are similar t-SNE plots where each dot's a cell once again, and they're colored by their cluster ID. And there's just 3 examples here. And what we observed was that the most common number of clusters within these glioblastoma stem cells was 2, but then the maximum can actually go up to 6, which we show here with G620. So this was really interesting to us, as we thought previously that maybe we were just growing out a bunch of cells that looked very similar to each other. But here, in fact, we have evidence that there are different transcriptional programs that are predominant in different groups of cells within one culture. So next we wanted to ask if these clusters that we find within patients have similarities across patients. So to do this, we took the marker gene list for each of these different transcriptional clusters, and we simply compared them to find which clusters were most similar across patients. Perhaps unsurprisingly, what we found was that the most commonly shared marker genes are related to proliferation. So we have some really canonical cell cycle genes like Ki67 and TOP2A showing up here. So then next we wanted to ask if these different transcriptional subclones that we see in the dish, kind of what distinguishes them. We know that in some cases it's marker genes, but we wanted to know if in other cases perhaps it was due to genetic variation. So to answer that question, we inferred copy number variation for every cell within the sample and then looked at the average across the cluster. So in this plot here, each column is a different chromosome. Individual GSC samples are separated by dark black bars, and then within each of these dark black bars, the kind of mini rows represent the copy number variation for each cluster in that sample, so the different colors on the previous slide. What we can see in some cases is that there is quite a bit of genetic variability within individual samples. So the sample I highlighted previously that had 6 different clusters, you can actually see here that there's quite a bit of copy number variation that differs between these clusters. So in some cases, 2 of the clusters here have a deletion of chromosome 6. Same thing goes for 9, where there's variable alterations across that. And also 4 of these 6 clusters have a deletion of chromosome 10, whereas 1 does not. This is the case for several other samples, including G637 down here, where we see that deletion of chromosome 10, a really classic GBM CNV, is private to only 2 of the 5 clusters in the sample. So what this tells us is that in addition to variation due to marker gene expression, there's also copy number variation going on here within these different cells. So next we wanted to ask that we kind of understood what drives heterogeneity within individual GSCs. We wanted to understand what's driving this heterogeneity across all GSCs, and if we can summarize that into kind of an interpretable model. So to do this, we ran PCA on our collective population of all cells, so just over 69,000. And what we found was that the first principal component actually separated these GSCs into 2 groups. So the first group, which roughly correlated to being PC1 high, was upregulated for genes that related to inflammation, a bunch of marker genes, epithelial to mesenchymal genes. And also in comparison to cell types in the normal brain, it looked like A2 reactive astrocytes, as well as cultured astroglia. Whereas on the opposite end of principal component 1, so PC1 low, we saw that all these GSCs were enriched for kind of neurodevelopmental genes, and they also looked like a collection of cell types that resemble fetal brain cell types. So the interesting part to this analysis was that it wasn't as if each cell was kind of one or the other group. There was kind of this gradient between the 2 extremes. So based on the pathway analysis, similarity to normal brain cell types, and also marker genes that we saw being expressed, we turned the PC1 high GSCs as being injury response-like due to their kind of wound response programs being expressed, and then the PC1 low being developmental due to their similarity to neurodevelopmental programming. So to kind of corroborate these results in a larger cohort, we performed bulk RNA sequencing on 72 GSC lines, and we found that the optimal number of clusters was 2, and then we did pathway analysis on those 2 different clusters found from bulk RNA sequencing. We found the same pathways were showing up as in the single cell RNA sequencing. So there was one group that was really high for like immune and cytokine and kind of injury response related pathways, whereas the other was much higher for neurodevelopmental related pathways. So then we want to ask, when you look within each GSC culture, do you see cells from kind of both ends of this spectrum, or is it mostly just one or the other? So here we have violin plots, where in red we have the gene signature score for each cell from that sample for the developmental program, and the black ones represent individual gene signature scores for the injury response program. And then each kind of column here represents a different GSC culture. And what you can see is that this gradient still coexists, and it's perhaps more prominent in this visualization, where you can see that there's sort of patients that kind of fall along this scale of the gradient. So some patients are very high for the developmental subtype, very low for the injury response, and vice versa. And then in the middle here, we have these samples where they kind of have a mix of both subtypes. So at the bottom, we visualize this relationship using two axes, where the x-axis represents the gene signature scores per cell for developmental, and the y is the injury response. And you can see in some cases, all the cells look developmental. In others, all of them look injury response-like. And then you have some samples here in the middle where you kind of have this moderate mixing phenotype. And then in some cases, you actually have, such as G549 here, the majority of cells looking injury response-like with rare populations of developmental-like cells. So next, to help us interpret what this gradient that we identified in the transcriptomics data means, we collaborated with the Angers lab at the University of Toronto with his really great postdoc, Graham, to help us do some genome-wide CRISPR screening in a collection of 11 GSC cultures. So to do this, we use the Toronto knockout library, which is basically a genome-wide knockout screen to help us identify which genes are essential for proliferation or kind of functionally essential within these different cultures. So we identified a list of essential genes for each sample, and then we just did a simple sample-to-sample correlation to help us identify groups of samples with the most similar gene essentiality profiles. And what we found was that the gene essentiality profiles formed two groups, and then when we compared that to our transcriptomic scores for the developmental and injury response phenotypes, it actually overlaid quite well. So we found that in addition to the transcriptomic data identifying these two GSC states, it also is read out in similar ways using our functional data with CRISPR screening. So next, we wanted to specifically identify which genes are essential within the developmental or injury response groups. So to do this, we took the basically the differential essentiality analysis, and we found lists of genes that were essential in either injury response or the developmental group, but not in both. And similar to our previous analyses, found that the injury response GSCs are dependent on genes related to inflammation, integrin signaling, whereas the developmental GSCs are more dependent on genes related to neurodevelopment, as we would have expected from our transcriptomic results. So these include genes like OLIC2, SOX2, and ASCL1. So next, we wanted to see if we compared this functional data kind of across the sliding window, how does it relate to our gradient? Do we still see these samples that are in the middle of our transcriptomics gradient having kind of middler phenotypes with the functional data? So here what you can see is each sample as the columns, and then we have for each sample what proportion of its essential genes are either specific to injury response or developmental programs from the left side of the slide here. What we can see is that samples that are scored very high for the injury response phenotype with transcriptomics have the majority of their essential genes corresponding to the injury response phenotype from CRISPR. But then as you kind of move along this gradient and you get into this middle section where we have these two samples, especially G809 that falls in the center of our transcriptomics gradient, you can see that it's a lot closer to being 50-50, where it's reliant on a mix of essential genes from both the injury response and developmental phenotypes. So finally, now that we have these two groups that we identified in the glioblastoma stem cells, we wanted to compare this to see how this carries over directly into patient tumors. So to do this, we sequenced 7 patient tumors, which totaled just over 44,000 cells. So of these 44,000 different cells, about 14,000 of them were malignant. And to identify these malignant tumor cells within our single-cell RNA sequencing data, we used a kind of 3-pronged approach, where the first is clustering. So we're basically looking for co-localization of similar cell types, knowing that these tumor cells probably aren't going to overlay properly because they have very different transcriptional programs. In combination with the clustering, we also just use a panel of marker gene expression to help us figure out which cells are immune cells and which cells are tumor cells. Then we use gene signature scoring to basically nominate which cells are of brain origin and which cells are of immune origin. Then using this information, we gate out all the brain-like cells or possible brain-like cells. So this includes normal and tumor cells, and we feed that into a series of different copy number inference tools to help us identify which of these brain-like cells has different copy number variations that we would expect to see in GBM, such as gain of chromosome 7 and loss of chromosome 10, which is indicative of being tumor-like, and then which ones do not have these alterations, which would help us understand that they're normal cells. So using that analysis, we take those 14,000 or so malignant cells across 7 tumors, and we ran PCA again with all of our GSCs. And what we found when we interpreted the first 2 principal components, there's actually 2 linear programs that are explaining the bulk of variance in this data set. So the first one here, shown in red, is what we call the differentiation trajectory, where you can see that there's this kind of movement from the GSCs down to more differentiated tumor cells. And we look at the genes that define the different ends of this program, we see that the GSCs, they look a lot more similar to young astrocytes, whereas the primary tumor cells resemble more mature astrocytes. And then secondly, we went to analyze this kind of second axis we see here. And what we found was that it was actually the same transcriptional gradient that we found previously in the GSCs, which explains, which shows that the GSCs go from this developmental to injury response state, which I talked a lot about earlier in this presentation. But then also we kind of see that these primary tumor cells also express these inherited phenotypes from the GSCs, where they also look like the developmental or injury response-like cells. So finally, we wanted, in collaboration with Gary Bader's lab, we wanted to do a more formal identification of which tumor cells actually look most like the GSCs. So to do this, Gary's PhD student Owen helped us with a logistic regression classifier, which will basically help us nominate GSC-like tumor cells from our patient tumor single cell data. And perhaps unsurprisingly, what we found was that the cells that looked most like GSCs in the tumor were in this region of overlap on the PCA plot. And then we do differential gene expression analysis between these GSC-like tumor cells and the more mature-like tumor cells that would fall down here in this black tail. We see that these mature-like tumor cells, they overexpress genes like GFAP, aquaporin-4, and CLU, which are all markers of astrocytes, which goes with the gene signature scoring analysis that we saw in the previous slide, whereas these GSC-like tumor cells are upregulated for more neurodevelopmental genes, such as SOX4, SOX11, and then also proliferation genes. And furthermore, we found that these GSC-like tumor cells, they have higher expression of young astrocyte gene expression programs, supporting perhaps that they look more like an earlier developmental state, whereas the mature tumor cells that fall in this black tail look a lot more like mature astrocytes. So in summary, we proposed a model whereby transcriptional heterogeneity in glioblastoma can be explained by a combination of 2 different gradients. The first is a glioblastoma stem cell gradient between regenerative and wound response programs, and the second is a bulk GBM gradient between these stem-like and differentiated cells that mirror astrocyte development. We validated our developmental to injury response gradient using both bulk RNA sequencing and identified functional vulnerabilities using genome-wide CRISPR screening. Using this CRISPR screening, we revealed some dependencies that could represent potential therapeutic targets, specifically tailored to GSCs within GBM. And we also observed a high degree of transcriptional heterogeneity within individual GSC populations, which can be partially explained by cluster-specific copy number variants. So moving forwards, we are hoping to validate some of these findings directly in tumor tissue using some wet lab methods, and then also to kind of up our patient tumor number by using some single cell or single nuclei RNA sequencing. So with that, I just wanted to thank everyone for listening and thanking the organizers for giving me an opportunity to present this work at this conference today. And of course, this is a huge collaborative project with lots of different groups contributing, especially Owen from Gary Bader's lab, as well as Graham from Stefan's lab. So thank you very much.